So we know, and at this point, I think this slide is very well known, that we have we are going through a difficult pandemic, and that has devastating consequences. And of course, SARS-CoV-2 is the virus that induces COVID and can infect everyone. However, pregnant women are more susceptible to get admitted to intensive care unit. They had more documentation of mechanical ventilation, and importantly, there are display high rates of mortality. And all this data was recently reviewed by the CDC. In addition, pregnant women are at increased risk for pregnancy complications such as preeclampsia, stillbirth, and medically indicated preterm birth. And preterm birth is the leading cause of neonatal morbidity and mortality worldwide. So SARS-CoV-2, this is one of the reasons why we started investigating the effects of SARS-CoV-2, because our branch is focused on finding ways to prevent prematurity. And early in the pandemic, one of the main concerns of pregnant women was, if I am infected, can the virus infect my baby, the fetus, and in this case, the placenta? So this is the main question that was raised, because very early in the pandemic, it was reported that a newborn was infected with the virus. This was a 6-month newborn, and therefore it was unclear whether he was infected in utero or after delivery. Later on, there were other clinical studies showing that neonatal infection is possible. And in this study, they evaluated 33 neonates. However, most of them were negative at the time of testing, and only a few of them were positive. I'm not going to go into details in this study, but what they found really is that the only very few neonates were positive, and however, the testing was done on day 2, which raises the possibility that the infection was not in utero, and it may be that was acquired postnatally. However, soon after, another paper came out showing that, claiming evidence of vertical transmission. And this was a case report of a woman who presented at 32 weeks of gestation. Term deliveries occurred after 37 weeks of gestation. Therefore, this was a preterm gestation. She presented with several symptoms that led the physician to think that she may be positive for SARS-CoV-2. She tested positive for SARS-CoV-2. She developed pneumonia, and then she was treated with a cocktail of antibiotics. Because she was in preterm gestations, and one of the fears at this gestational age is that maybe the woman will start having contractions. The physicians decided to test the amniotic fluid, which is done through an amniocentesis, and it's a procedure that is semi-invasive. And the amniotic fluid is surrounding the fetus, and therefore is considered the fetal compartment. So this tested positive for SARS-CoV-2. However, the neonate tested negative at birth, and then it turned out to be positive after 24 hours. So it was a little bit of confusion, but this was the first evidence that the virus could gain access into the amniotic cavity. Unfortunately, the mother died in the postpartum period because of the severity of COVID. So this case report was very important because it raised the possibility that the virus in severe cases of COVID can reach the fetus. However, if the virus reaches the fetus, then the fetus will create a humoral response, which will be characterized by the increase in levels of IgM. And of course, the IgG will be also increased because the mother will transmit those immunoglobulins. So in this study, they evaluated the IgM and IgG levels against SARS-CoV-2. However, among 6 of these neonates, only 2 of them were tested positive for IgM specific for SARS-CoV-2. However, the neonates born to these women with COVID, and it was a severe COVID, they tested negative, didn't display any symptoms of COVID, and tested negative for SARS-CoV-2 in the serum and the throat swabs. Therefore, there was still a polemic about whether high levels of IgM were indicative of infection because the neonates tested negative, and also they didn't display any symptoms. And I must add that most neonates born to women with SARS-CoV-2 infection and severe infection are delivered at term. And therefore, most of them, I would say in general, do well. So soon after, a year later, after all these case reports and different evidence of vertical transmission, a group of investigators decided to systematically review these cases, and they concluded that vertical transmission was very rare, only occurred in 3% of the cases when they summarize most of these cases. So therefore, vertical transmission of SARS-CoV-2 is possible, but it's rare. And it seems to be exclusively seen in cases with severe COVID. However, can the placenta be infected? Because the placenta has a protective role, and therefore we don't know whether the virus is going, in this case, if we found the virus in the amniotic cavity, it seems that the placenta may be infected, but there was no evidence of placental infection at that point. So there were some early studies indicating that the placenta, and the placenta has two main structures, you may say, the placenta itself and the membranes that surround the amniotic cavity, that is, they are an extension of the placenta and what we call fetal membranes, chorioamniotic membranes, or extraplacental membranes. So this virus was detected in the placenta and the extraplacental membranes in only 3 cases, and none of these out of 11 that were evaluated. However, none of the infants that had or tested the placentas tested positive for SARS-CoV-2 tested positive for SARS-CoV-2 themselves. So that again tells you that the placenta can be infected, but maybe the fetus was not infected. Later on, a little bit with more sophisticated techniques, researchers demonstrated that the virus was present in the placenta. And this is, for those who are not familiar with the structure of the placenta, this is a slide representing the villi that is immersed that connects the maternal and the fetal circulation. And what you can see here is that the majority of the staining is in the areas that are surrounding they are establishing the interface between the mother and the fetus. The fetal blood will be in this compartment, and the maternal blood, or in the intervillous space, will be in this part. So what is here showing you, this will be the maternal, and this will be the fetal. That's what this picture is showing you, is that the virus is restricted to the maternal side, but yet it is present in the placenta, and this was confirmed by in situ hybridization. Later on, there were a group of investigators demonstrating that, suggesting that vertical infection of the placenta has to be documented by using different techniques. And recently, the NICHD hosted a workshop in order to establish new criteria, and these criteria are very rigorous. So the degree of infection of the placenta may be actually lower than what is reported here. It's still rare. It was considered that only a few cases that they tested positive for SARS-CoV, mothers that tested positive for SARS-CoV-2, their placentas were positive for the virus. Therefore, placental detection of SARS-CoV-2 is rare. And I mentioned placental detection of SARS-CoV-2 rather than placental infection, because infection will mean that the virus or the microbe is detected, and also inflammatory responses are triggered. And at this point, nobody had documented that. However, as I mentioned earlier, placental invasion, or the fact that the virus is present in the placenta, doesn't mean fetal infection. Fetal infection will mean when the fetus is triggering an inflammatory response, and the virus is detected in the fetal organs. And that doesn't happen very often with fetal infection, because the placenta has a very important role in protecting the fetus against infections, and not just against SARS-CoV-2. This has been widely investigated by many investigators, specifically Dr. Caroline Cohen. She has done excellent research showing that the placenta and the syncytial trophoblasts, which are these finger-like structures, are very good at protecting the viruses in the maternal circulation, invading the fetal circulation. So therefore, the placenta is a very important organ and is an immunological barrier. And because we did, at this point early in the pandemic, we didn't know whether the placenta was being infected, and therefore the fetus was being infected, we decided to investigate the mechanisms of cell entry of the SARS-CoV-2 virus in the placenta. And we know at this point that SARS-CoV-2 utilizes different mechanisms, but it is mainly utilizing ACE2, which is the receptor, and some proteases in order to infect the cells. So the ACE2 receptor, and in this case, the TMPRSS2. And of course, this audience is very familiarized with all the mechanisms of entry. We focus on this mechanism of entry because it was at the beginning of the pandemic was the main route of cell entry for SARS-CoV-2. So it's required that ACE2 and TMPRSS2 are co-expressed in order to make it the canonical entry of SARS-CoV-2 occurs. And we know that ACE2 is expressed, of course, in the lungs, intestine, and liver, and so on, and is highly expressed. And because of that, we display the symptoms when we are infected with SARS-CoV-2. However, at this point, there was no research performed in the placenta. So that was our question very early in the pandemic. Can the placenta be infected by SARS-CoV-2? So we did some investigation in this and was reported in this study. And in a nutshell, what we did is to take tissues to take samples from the placenta. This is the placenta. These are the extraplacental membranes, what I was mentioning to you, and this is the amniotic cavity. So because we don't work with first trimester tissues or fetal tissues, we could utilize some available data from the UK, and here they characterize utilize single cell technologies in order to characterize the single cell dynamics of the placenta in early pregnancy. And then we also collected a sample from second trimester that came from a woman who had a pregnancy complication that is called a placenta creta, which leads to the excise of the uterus, and therefore we were able to collect the placenta at this gestational age. And most of our research is done in the third trimester because this is where the pregnancy complications, the onset of pregnancy complications occur. And we are capable of collecting placentas in a very rapid manner. So we had already published at that point that the single cell signatures of the placenta in preterm and third parturition. So we utilized this data that was available for us, and also the first trimester data. We generated new data in order to see whether the placenta was expressing these canonical receptors throughout gestation. We also evaluated the choriamniotic membranes because I was mentioning earlier, the choriamniotic membranes are very important immunological barrier that in cases maybe that the placenta is not, the virus doesn't come directly from the placenta, maybe the virus can also come through the myometrial tissues. So we evaluated all those. And what we did is, in a nutshell, we did some single cell transcriptomics, and using computational methods, we categorized different types of cell clusters. And the major cell clusters in the placenta are trophoblasts, and these are the cells, the fetal cells that create the placenta, stromal cells, myeloid cells, lymphoid cells, and endothelial cells. And of course, they have different types of names, but I don't have time to go into the details, but I really would like to explain a little bit more about the trophoblast. So the trophoblast, there are different types of trophoblast, and this is the maternal side where the myometrium is. Something that we call the placental bed is where the placenta rests throughout gestation. And what happens is that in the placenta, you have this layer, and what I was showing you earlier, there here, these finger-like projections, and all these intervillous space is maternal blood, and everything that is yellow here is syncytial trophoblast, which is a multinucleated trophoblast. And this is covering and very nicely the fetal compartment. This is fetal blood, and therefore there isn't communication between the mother and the fetus, yet there are different exchanges of nutrients and gases and so on. So this syncytial trophoblast maintains this separation between the mother and the fetus. So because it's a multinucleated cell and single cell transcriptomics, it's mainly focused on cells that can be encapsulated in the droplets. And we wonder whether we have enough syncytial trophoblast, which will be the main barrier here protecting the fetus in our samples. And we can see here that the syncytial trophoblast, because of their multinucleated and large size, is not really efficiently captured in single cell technologies. However, we do have syncytial trophoblast, extravillous trophoblast, and actually we discovered a new type of trophoblast, which we call non-proliferative interstitial trophoblast. So we have different types of trophoblasts, and also syncytial trophoblasts, because of the digestion procedures that we utilize, is more likely to become one of these trophoblasts. So we still have trophoblasts, but we had to have in consideration that the syncytial trophoblast was underrepresented. To alleviate this concern, what we did is utilize single nucleus RNA-seq. So instead of encapsulating cells, we extracted the nucleus, and nucleus were processed in the same way than the single cell suspensions. And here we can see that in the single nuclear preparation, syncytial trophoblasts were enriched. So therefore, we had single cell data and single nuclear data. So the results were that when we evaluated the co-expression of ACE2 and TMPRSS2 in the first trimester placenta, we find that the expression, and you can see only a few dots, the co-expression of ACE2 and TMPRSS2 were negligible. So we're very, very minimal. In the second trimester, the same. Very little co-expression of these two. And when we analyze in the third trimester, where we have more placentas that were isolated by single cell and single nuclear RNA-seq, we can see that the co-expression of ACE2 and TMPRSS2 was also very minimal. In the membranes that are surrounding the amniotic cavity, we also didn't find the co-expression of ACE2 and TMPRSS2. So it's, I just wanted to clarify that we are talking about the co-expression of ACE2 and TMPRSS2 rather than the co-expression of ACE2 alone, which is known to be expressed by the placenta. So because we didn't find the expression of the canonical receptors for SARS-CoV-2, we first wanted to know whether other receptors from viruses that we know that they cause congenital disease, such as cytomegalovirus and Zika virus, can be expressed by the placenta, because it may be that some of our pipeline or some of our methods may not be ideal for the exploration of canonical receptors. So what you can see here is that the placenta, different types of the placenta in different types of first, second, and third trimester, they do express the receptors for CMV and as well for Zika virus. And these are some of the receptors. More information is in the paper. And so therefore, what we concluded is that indeed this SARS-CoV-2 canonical receptors were not expressed by the placenta, but the placenta, we could find the receptors for other viruses such as cytomegalovirus and Zika virus. However, there is a possibility that the placenta is expressing non-canonical cell entry mediators for SARS-CoV-2 infection, because we know that it is possible even though it's rare. So we evaluated the expression of other transcripts, and here you can see that indeed the placenta can express all these receptors, and also we have more information in the supplementary material. But you can see here that compared to the canonical receptors, there is little expression of these canonical receptors. Therefore, from this study, we concluded that the human placenta does not highly co-express the canonical receptors, ACE2 and TMPRSS2 for SARS-CoV-2. However, it can express receptors for other viruses that can cause congenital disease, and can also express a non-canonical receptor for SARS-CoV-2 infection. However, the fact that the receptors are not present in the placenta, and we think that this is a mechanism whereby the placenta is protecting the fetus against SARS-CoV-2 infection, that doesn't mean that the SARS-CoV-2 infection during pregnancy in the mother can not have effects in the fetal life. And because of that, we performed a second study in which we evaluated the maternal and fetal inflammatory responses triggered by SARS-CoV-2 infection. And this was reported here earlier this year. And what we did in this study was to recruit women who tested positive for SARS-CoV-2. Most of them were asymptomatic because most pregnant women don't display symptoms like the general population, and very few of them were severe. And we compared these pregnant women with women who tested negative for SARS-CoV-2, and they were gestational age match. So because at this point we didn't know what we were looking for, we know that the placenta doesn't express the receptors, but we don't know what are the main targets of SARS-CoV-2 during pregnancy. We utilized a multidisciplinary approach that included immunology, single-cell biology, computational biology, placental histopathology, and microbiome approaches. First, we detected IgM and IgG in the maternal blood. And what we found is that indeed mothers that tested positive for SARS-CoV-2 have high concentrations of IgM and IgG as expected. However, in the cord blood, we didn't see an increase in IgM, but they do have an increase in IgG, which is transferred from the mother to the fetus. So this data show that the fetus itself maybe is not creating an humoral response at this point of infection. Therefore, we next analyze cytokine responses because the fact that doesn't trigger a humoral response, it may mean that this that doesn't mean that it's not responding. The immune system of the neonate is not responding. So we analyze the maternal blood, and these are, because most of these women are asymptomatic, but we see that there is an increase in interleukin 8, interleukin 10, and interleukin 15. And actually the severe cases, those 3 severe cases didn't drive these cytokines. Actually, they were pretty mixed among the between the asymptomatic and the symptomatic. So the mother has an inflammatory response that is characterized by the increase of interleukin 8 and other cytokines, but it's not cytokine storm that similar to what is observed in severe patients. And in the cord blood, we find an increase in interleukin 8, which raises the possibility that this interleukin 8 was transferred from the mother to the fetus, or the fetus itself was producing interleukin 8. This is something that requires further investigation.
Nardhy, you have 2 minutes.
Thank you. So, and the next thing we did is to do immunophenotyping of these cells. And what we found is that with the different types of cell types, and you can see here in this heat map representation, is that there are differences between women with SARS-CoV-2 infection or controls. And we focus on T cells because this is some interest of the laboratory. And what we found is that the mothers have reduced T cells in the circulation, but fetuses didn't. And I just going over quickly about this slide, just showing you that there are specific cell type T cell subsets that they are decreasing in the mothers, but they were not decreased in the fetus. And lastly, we did single cell transcriptomics in order to explore what are the changes in the cells that there are in the placenta. And for doing that, we again utilize single cell transcriptomics, computational biology, and also the maternal and the fetal genome in order to differentiate between fetal and maternal cells. And in a nutshell, what we found is that the maternal T cells in the maternal fetal interface are responding against the virus, and also fetal stromal cells. And later we say how bad it was this maternal T cell activation. So we compared it with SARS patients with severe COVID that were hospitalized. And we can see that this is a positive correlation between these two transcriptomes. And by doing a gene ontology analysis, we can see that the majority of these transcripts were involved in mitochondrial translation. And we also did RNA sequencing on the maternal and the cord blood. And this is just to show that the mother is responding to the virus, but also the fetus is responding to the virus in a milder way than the mother. And last thing that we did is to compare our placental signatures to our maternal and cord blood signatures in order to see whether we could not invasively detect the changes in the placenta in the mother or in the cord blood. And as we can see here, we can identify the changes for T cells and macrophages in the mother, but we couldn't detect the changes in the fetuses. And we did a bunch of research just to show that the placenta is not, we were not able to identify the virus in the placenta. And we also did more research in showing that the virus cannot be identified in, cannot alter the sterility of the placenta. So in conclusion, we found that the mother creates an inflammatory response, but the fetus also increases, displays increased levels of interleukin-8 without the detection of IgM. There are alterations in the mother in the T-cell repertoire that can be associated with those patients with severe COVID, and these signatures can be detected in the maternal circulation. However, the virus cannot be detected in the placenta in our cases, and it didn't compromise the sterility of the placenta. So this is one of the research that we have done, and we are currently investigating what drives this susceptibility. And we have documented some of the research here because we know that pregnancy is a unique immunological state, and we had already documented that. And just in a nutshell, I can tell you that pregnant women are way different. Their immune system is very different and responds very uniquely to the viral particles as well as viral proteins. And lastly, I just would like to thank my team for the people who do the work, Valeria Garcia, who was responsible for the characterization of immunological characterization of COVID patients, and of course to our collaborators and our funding sources. And I'm happy now to take your questions.
Well, thank you, Nardhy, for a fascinating talk, a beautiful presentation. Now open for questions. Let's see, we have one here from Morgan Firestein in the Q&A. Thank you for excellent talk. Can you please confirm whether the participants had an active infection at the time of sampling? And if not, did the amount of time between infection and sampling impact the level of inflammatory responses?
Yes, that is a very important question. And all of the patients that we call SARS-CoV-2 positive patients were patients who were admitted in the labor and delivery unit because of the symptoms. So all of them were undergoing different symptoms. How long do they have the infection? Well, that will be difficult to track. But the majority of them display symptoms, and this is when they were tested positive, and therefore they were included in our study.
Okay, and I have a question. You may have addressed this earlier in the talk, but do you have any information about the antibody status of the offspring of the mothers who were infected?
Yes, this is a great question, and this is one of the main concerns after we documented that there is fetal inflammation, and whether this inflammatory status or even the immunological humoral response is detected in the newborns. However, unfortunately, we couldn't follow up the newborns, the infants, and this is something that we are currently doing, trying to investigate whether those effects in the fetus have deleterious effects, not just in the immunological memory by IgM or IgG, but also whether they have other alternatively devastating effects in the neonatal period.
Great. Can you unshare your slides? Are there other questions? Eric, did you have a question?
Yeah, just very quickly, Nardhy, that was a great informative talk. You had indicated early on that up to sort of like 4 or 4.5% of the placentas you detected virus in. And I'm just wondering, how do you think the virus got there if there are no receptors for the virus in the placental tissue? Yes,
I think the virus can be there because I don't think it's entering into the cells. I think it's accumulating because most of the evidence has shown that the virus is restricted in the syncytial trophoblast. And it doesn't look like the cells are, it's more like accumulating from the maternal side to the fetal side. And it's very clear that the fetus itself is not being infected because it's restricted in that maternal compartment. Now, the SARS-CoV-2, the placentas that we included in this study were normal deliveries, normal pregnancies. It has been shown that the infection of SARS-CoV-2 can upregulate the expression of these receptors, but these studies, of course, have been done in vitro. So whether that happens in vivo, we don't.